right. I think we're live. Don't forget homework due today. I put the due date this afternoon. So I can help you knit stuff in the afternoon. We'll have lecture. We'll continue this probability. Our exam is on Monday next week. Okay, we'll do it here. I think we'll do it during lab. So starting at 2, we'll take the exam. It's in 2 parts. We'll do the closed book part, and then you have the application part, which is an RMB file. All right, with the RMB file, I think we'll just upload it. Save on printer paper. Don't have to worry about the printer actually working or having a staple. Yeah. All right. Ready? I guess we can take questions for homework, or we can just wait for the homework questions until after our lecture at 2. Good. All right, so we're going to introduce probability. Why probability? Because that's what stats is. We're coming up with a probability that our data, that we would get the data that we actually obtain if our null hypothesis is true. All right? And for all of our null hypotheses, it's going to be like no difference. There's no groups whatsoever. All of our means are equal, variances are equal, and so forth. So what we're going to do is we calculate the probability, and if the probability is low, then we reject the hypothesis and conclude, okay, something other than chance produces these differences. So it's all about chance events, and we can model chance events using probability. So in the OpenStax book, this is part of chapter 3. There's a lot of material that's out there. If you don't fully understand some of the stuff, that you can ask me questions, or you can try to find some stuff that's online. All right, so probability is the foundation of all statistics. We're going to perform experiments, make assumptions, and then we compare our data to what we would expect based on those assumptions. You've already probably done this using chi-square tests. I know you did this in intro bio. I know you learned the chi-square test in intro bio. You might not remember it, but you collected data and you compared it to a predicted frequency. Most likely scenario is you did it using Punnett squares to generate your expected. All right. Now, what happens when we deviate from our expected? Well, when you deviate from the expected, that usually leads to some important biological phenomena. Why do we care about the deviations? Because when we make our hypotheses, we're generally doing these hypotheses, these experiments, aimed at rejecting the hypothesis. All right? We can't prove it, but we can reject it. We can say, look, chance did not produce this outcome. All right? So what we use is something called a p-value. You're going to know this definition backward and forward, all right? Because it appears on multiple tests. So what's a p-value? It's a probability of obtaining a value or data as extreme or more extreme than our observed value or data if our null hypothesis is true. All right, so we're calculating probability. That's what a p-value is. Low p-values, our data are going to appear different from random, or at least chance the probability that random chance produces that is going to be pretty low. So what is probability? It's the likelihood that an event can happen. And normally, the probability that an event happens is equal to its frequency in the population. Right? So whatever the frequency is in the population, the probability that we would get that one item is equal to its frequency. Here's an example. We have a jar of marbles. All right, we've got 200 marbles in there with 4 different colors. We've got 120 red, 40 blue, 36 white, 4 green. All right, the probability that we would reach in and pull out a red marble is equal to its proportion in the population. So it's 120 red marbles out of 200. That's 60% of the marbles are red, which means that the chance that we pull out one red marble is 60%. Right? Blue constitutes 20% of the marbles. Probability that we pull out a blue marble is 20%. White's 18%, so it's 18% chance of pulling out a white marble. And the green is right at 2%, which means we have 2% chance of pulling out a green marble at random. Now the expected value is associated with the probability. An expected value is a weighted average of all possible values. That's what it is. What we would do is multiply the probability times the number of events we're going to have, which gives us then what we would expect based on that probability. So the example here is we have 400 marbles, all right? We're going to draw 400 times from that bag, all right? So if we draw 400 times and the probability of getting green is 2% for each draw, you're going to have 400 times 0.02, which means we're going to expect to have 8 green marbles in that sample. All right, so out of our 400, we'd expect 8 green marbles. Are we going to exactly get 8 marbles? No. Sometimes we'll get more, sometimes we'll get less. And that's because of stochasticity. Every time we draw a marble, we have an element of chance associated. So here's an example. This is going to use R. All right, and we can demonstrate this. So this is what I received when I did this, I don't know, whenever I made this presentation. What happens now? So what we're going to do is utilize the R multinomial, the multinomial distribution. And what I'll do is I'll make a new R markdown document. You can carry notes in here, but this part is more about just learning probability, probably more just pen and paper. Probability. All right. So I'm going to utilize demonstration. Okay, random sampling demonstration. Right. Our multi-gnome, n size of the blob. N size of the blob. Okay. So our multinomial will generate a random sample of appropriate size given a specific set of probabilities. Now, the help file might be a little bit more descriptive on what it is, but a multinomial distribution is multi-nominal variable distribution. It's like a binomial distribution is 2 nominal outcome distribution. So if we have sex, male versus female, that is a binomial distribution. All right. Whether a successful mating event, yes or no. All right, that's a binomial distribution. If we do, let's say, multinomial cars makes, we probably have a lot of you all drive different cars or whatever. All right, the category is nominal, and you have multiple outcomes for that car make. All right, that's a multinomial distribution. All right, so what we can do is use this to generate a random sample of appropriate size. Now the size is basically how many are we drawing. All right, and the n is number of replicates that we're going to do this. So if I want to draw, let's say 50 times, so we're gonna do coin flips. Okay, we can do this with the binomial distribution, but I'm going to use multinomial distribution for this. I hope it's going to work. Coin flip, head or tails, is technically binomial, but we'll use the multinomial to demonstrate the function. All right. All right, so we're going to flip the coin 100 times. So we want to generate one sample. That's our n. Our size is 100. We're going to flip it 100 different times. And our prob will be, we'll say 50-50. So our probability needs to sum to 1. Or I think there's an option here, I don't know if multinomial will do this, where it takes the numbers, like the expected numbers, and converts into a probability. Chi-square distribution will do that. I'm not sure if multinomial will do that. So I'm going to run this and see if that works. Yep. This function generates a single sample where we flip the coin 100 times and recorded the number of heads and tails. All right. The probability of each is 0.5. So this didn't really give H or T, but it gave a vector of 46 and 54. All right, and these numbers lined up with the order of our probability. So if probability of flipping a head is 0.5, probability of flipping a tail is 0.5. And in this 100 sample, we had 46 heads, 54 tails. Now we expected 50-50. Did we get it? No, we didn't. It was close, but we didn't get exactly 50-50. Why? Because of random chance. So what did you get?
50-50.
You got 50-50. There's one 50-50. Did anyone else get 50-50? All right, there's a second 50-50. I mean, it's possible. It's just random chance. All right, so this is our function. So what I'm going to demonstrate is this marble example. All right. All right, so this is our marble demonstration to demonstrate the effective sample size. All right, so what we're going to do is run 100 marbles, or a sample size of 100. All right, so our multinom, all right, we want one sample. Our size is 100, so we're going to make 100 draws, and this will be sampling with replacements. We're going to draw a marble and then we'll put it back. We'll draw a marble, put it back. All right, and our probability will be that 0.6, 0.2, 0.18, and 0.02. All right, so that was the probability that was on that table. Our imaginary bag of marbles, 200 marbles, that's our frequency. All right, so this will be 100 draws from the bag. And 500 and 1,000. All right. So what we're going to focus on is the green marble. All right. How many green marbles do we get? So when I drew it, we have 100. There's 2%, so 4 marbles total in the bag. Is that right? Yeah. 4 marbles total in the bag. So out of 100 draws, I would expect to see 2 marbles. I get 2 green marbles. How do I know this was green marbles? Because I had ordered our probabilities based on the color. So red was the 68. I think blue was the 20%, and white was the 18%. I think that was the order, and green was the bottom. What did you get? Did you get any green marbles?
I got 1.
You had 1 green marble. You had 4 green marbles. What about everyone else? 0. You got 0, 3. Did anyone have like 8 or 6? All right, let's up it. Let's go to 500 draws. Oops. 500 draws from the back. All right, so our multinom. I'm actually just gonna copy and paste this and change size to 500. All right, so now we have 500. I think we're expecting 6 marbles total. 6 green marbles, I get 8. What did you get? Anyone get like
10.
2? You had 10?
13.
13?
17.
Holy cow, 17? 15? Okay. Alright, let's do 1,000. See what we get. Alright, so I do 1,000. I get 7. I should have had almost twice the number than that 500. 1,000. There you see, it's the more we get, the more likely we're getting closer to our expected value. But everyone's getting a different value. So what I could do is run this script, this R multinom, 1,000 different times, and save how many green marbles I have. So now I have 100 replicates of these draws. All right. And what we see is that as we increase the number of our draws, our actual estimated probability, or the variation in that probability of getting a green, is going to get closer and closer and closer to that actual value. So in my 100 trials with 100 samples, I can calculate my probability is going to be anywhere from 0 to 8. When I ran this, so I had from 0 to 8 marbles in that sample, which is, pretty wide. Standard deviation is 0.01. Once I upped it to 500, we started to kind of temper some of the randomness, or you can say the average draws, the average frequency starts to minimize the effect of those outliers. So now our probability is going from 0.004 to 0.042. It's smaller than this, and our standard deviation is actually smaller. And when we get to 1,000 draws, this is at our smallest sample. So as sample size, as our number of replicates start to increase, the probability that the event will happen starts to approach its true value. All right? If we do it one time, we could be anywhere. But as we start increasing the number of replicates that we have, the average number starts to focus in and narrow in on the actual true value. All right? So we get closer to the true value, and dispersion around that mean also decreases. All right? So we can take this into account using standard probability theory. Now, what's important is when we do this, we're going for that random sample. We need a true random sample to minimize any bias that we see in our calculations. All right? So again, our goal is for unbiased sampling. If we get unbiased sampling, then the sampling frequencies in the data set should equal the true frequencies in our population. All right? And that means then, if we do that, then the probability of drawing any one item is the same as any other item. That's not to say that the probability of red is going to be the same as the blue. It's just the marble itself. Any one marble has the same probability as being drawn as any other marble, regardless of its characteristics. All right. Now, how do we collect the random samples? Just as a quick review, we've got 2 different ways. If we know the entire population, you assign numbers to them, and then you randomly draw their numbers. If we don't know the entire population, then we've got a couple different points where we can pick random locations where we start and then collect in those areas, or we can pick a point and just start wandering in various directions and so forth. So that was a quick review. All right, so random sample in R. How do we generate this? I'm going to show you how to do it. So we're going to load the Cricket dataset. Crickets.csv is loaded. All right, I want you to read it in. I ask you to read it in as dat.crickets. All right, we're going to demonstrate how we can get a random sample, how we can draw a random sample from a data set. This is a pretty good introduction for you, not because I think you'll actually have to draw a random data set, but this is a very good introduction to something called bootstrapping and randomization tests. All right, so I am going to get it in unmarked. So Crickets is posted. forget to use strings as factors. Make sure that's checked. All right. Did you get it? Did you get Cricket's data set? Should be easy for you to load in. If you struggle to load in this data set, you're gonna need to talk to me. We need to figure it out. You'll struggle on the exam, or at least the application part. So here's our data set. It's over. So here's the idea. We have 100 crickets. We have 3 different phenotypes in our crickets. I just have them, actually 4. C and D. There's A, B, C, and D. And what we want to do is some experiment using these crickets and having a representative phenotype in the experiment. So making sure that we have A, B, C, and D. Oh, I guess we also have E as well. Also E. And the question is, how do we go about randomly selecting that to ensure that we have all of these representative ones in our phenotype? Well, what we could do is go and individually number each of these crickets. And this is what I call the ID. All right. I did 4 digits. Who knows? You can do 3 digits if you want, just based on the experiment. All right. So what we want to do is we want to assess the susceptibility to infection. All right. But we don't want to favor any one phenotype. If we do this with, let's say, all of phenotype A, and they seem to be resistant to infection, you could incorrectly conclude that these crickets can't be infected, but that could be a phenotype A only. So, when we do the experiment, we realize we can't do all of the crickets. It's just too costly, time prohibitive, or costly in terms of time or money, or maybe some other type of stuff. So how do we randomly select 20 individuals? Well, to do that, we are going to use the sample function. All right. The sample function will draw a random sample from a vector that we give. So how big of a random sample? Well, we specify the size. If we ignore the size and just do sample of our vector, then it draws a random sample of the same size as our vector. All right. But then we have to change replace. So replace can be either true or false, T or F, capital. All right. So with the T or F, or the replacement, it's when we draw our sample and record it, do we put it back? Or do we pull out our sample, record it, and set it off to the side and say, okay, that's going to be number 1. That's what we're going to use. And then go back and draw it. So the difference is, like in our marble example, we sampled with replacement. So every time we drew, it was in a marble bag of 200, or you had 200 marbles. If you sample without replacement, every time you draw, you kept the marble out. So on the second draw, you only had 199, then you had 198, then 197, and so forth. So for a random sample, we don't want to draw the same cricket twice. Right? Once they get drawn, they're out. So what we want to do is sample without replacement. So here's our example for size 20. We're going to use sample. We're not sampling the data frame. We're sampling a specific vector. Oops. And what vector are we sampling? Well, that's the ID. We need to figure out what IDs are we going to use. So sample, X, size, and replace. We'll draw a random sample of size from the X vector. We can sample with replacement, So replace equals t, or without replacement. Right. Here, we'll draw 20 crickets without replacement. Generate our random sample of 20 crickets. So it's a sample. We've got that dot crickets, but that's the data frame. We have to give it a vector. And the vector that we're going to give it is ID, because that's how we know which individuals we have. So there's our numbered ones. We're going to take that vector, we're going to draw a random sample of it. We need size equals 20, and replace equals false. And when I generate, now I get a new vector of IDs. These are IDs. These are going to be the crickets that we draw. And we pull the random sample. So the probability of pulling phenotype E will be based on its frequency in our population. So if there's only one individual, and you've got a 1% chance of pulling it, chances are it's not going to be in this random sample. But if you have, let's say, 40% phenotype A, 40% phenotype B, then those two phenotypes will probably represent most of our individuals in this random sample. Now, every time I run this, I'm going to get a different random sample. All right? So, if you're going for a random sample, I might do something like that to save it. RID, so I save the vector, because then you can say, well, man, that's going to be kind of hard to get these numbers. Well, there's also a sort ID, a sort function that I can then sort them and say, oh, okay, good, they're in order now. All right. What about set seed function? Yeah, if you're gonna do this for like an actual scientific experiment, I would probably set the seed. Set the seed, run it, so that way, if you're in the middle of the experiment and your sheep is eaten by your dog, all right, your data sheet, you're like, okay, what was those ID numbers again? Well, you can go back and regenerate it using the set seed function. Did you save it first as an object RID? So I just kind of do the sample, save it as an object, and then I can sort it. Or you can use sort on the sample itself and automatically generate it. That's kind of like an FYI type technique. So, key points. X has to be a vector. It can be a numeric vector, it can be a list, right? It just has to be some sort of vector. That means then if you're working with the data frame, you're going to use your data $ variable name to get that vector. Number two, I do say don't set the seed when pulling a random sample. What we don't want to do is set our seed and keep using the same seed over and over and over again as we do our random sample. All right. When I do some of our bootstrapping, or if I do our randomization test, yeah, I don't set the seed. I just let it go unless I'm getting ready for a publication and I'm using it to generate images, figures. Then I'll set the seed to something, generate it, so if I have to remake that figure, I can get the same data. But for now, for like in your exams and stuff, don't set the seed. Any questions? All right, let's move to probabilities, working with probabilities. So we've introduced probabilities. All right, probability of drawing an event or drawing an item is based on its frequency. It's equal to its frequency. but we're normally not going in and drawing one item. We're normally going in and drawing multiple items, and that's the basis of our experimentation. You have a lot of different outcomes that can happen. All right. So how do we work with all of those probabilities? Well, we've got a couple of rules that help us calculate probabilities. The first one is the addition. The addition rule states that if we have two events, A and B, that are mutually exclusive, then the probability that one event will happen or the other is equal to the sum of their probabilities. That's what it is. Probability of A occurring or B occurring is equal to the probability of A occurring plus the probability of B occurring. Now, what does it mean by mutually exclusive? Events are mutually exclusive if they cannot occur together. All right, if they cannot occur together. So, an example of a mutually exclusive event is our daily lottery drawings, like pick 3 and pick 4s. Each ball is mutually exclusive of the other ball. Right? You can't have a ball get drawn out of there that is both a number 1 and a number 2. Right? So those events on a single ball, those are not mutually, or those are mutually exclusive. They can't occur together. But the balls on two different machines, they're not mutually exclusive. Now you can get a 1 on one machine and a 2 on the other machine. The other way to think about mutually exclusive events is let me come up with another example. I've got the examples here for the dice. Biology. Hair color. All right. You got brown hair, blonde hair, black hair, red hair. Right. You can't naturally have both brown and blonde hair. Catch the natural. All right. The hair color is mutually exclusive on an individual. But if you look at hair color and sex, now you have the events that are not mutually exclusive from each other. So if we ask, what's the probability that we select any individual here and they have either blue hair or blonde hair, we can go through and look at the frequencies and say, okay, the probability of getting a brown hair is this, the probability of getting a blonde hair is this. We add them together. That's our probability of getting brown or blonde. For dice, we've got 6-sided die. All right, so you can't roll a die, a 6-sided die, and get the numbers 1 and 6 rolled together. It's mutually exclusive. You roll once, you get one number. All right, but you can, on a repeated die roll, get multiple numbers, multiple sequences. So for our 6-sided die, if we ask what's the probability of getting a 3 or higher roll, we're asking the probability of 3 or 4 or 5 or 6. All right, those are mutually exclusive events, so we can just calculate the probability of rolling a 3, of a 4, of a 5, of a 6, and then add them together. So the probability of rolling a 3 is 1 out of 6. We got 6 options. All right, 3 is one of them, so 1 out of 6. 1 out of 6 for each of them gives us a total probability of 4 out of 6, or 2/3. 2 6-sided dice. Now, the numbers on those 6-sided dice are not mutually exclusive, but if we add their numbers together, now we produce a mutually exclusive event because you can't roll 2 dice together and get both a 7 and an 11 pop up. All right. But if we ask, what's the probability of getting a 7 or an 11? Again, probability of getting a 7, 6 out of 36. Probability of getting an 11, 2 out of 36. 3 out of 36. 2 out of 36. So now you've got 8 out of 36, or 2 nuts. 2 nuts. You'll get plenty of practice. So, events are mutually exclusive if they cannot occur at the same time. So you flip a coin, heads or tail, those are mutually exclusive events. If you draw a card from a deck of playing cards, getting a king or a queen, that's mutually exclusive. Sex of offspring, male versus female, those are mutually exclusive. clutch size, getting a 3 versus a 5. You can't have both 3 offspring and 5 offspring at the same time. Just they're mutually exclusive, as well as infection status. Then I asked, can you think of other examples that are mutually exclusive like this? Any? Give me one. What's that?
Eye color.
Eye color. Eye color.
Age.
Age? Yep. Yeah, you can't be both like 21 and 61 at the same time, right? All right. So I think mutually exclusive events, these are actually pretty easy to work with. And you typically do the probability of getting A or B usually falls under mutually exclusive events. Non-mutually exclusive events are those that can occur at the same time. So they can be 2 different items or characteristics, and they can be a sequence of events. That's normally what we're working on. All right, so 2 different items, characteristics, or a sequence of events. So we can look at sex of an animal and their infection status. Those are not mutually exclusive. All right. So sex and hair color, male, female, brown versus blonde, age and infection status. Rolling a paired out dice, the outcome on each, not sum together, but the outcome on each. Those are not mutually exclusive. Sex of offspring in a clutch of 4. All right, so if we do the number and the order, or the order, and then sex, those are not mutually exclusive events. All right, and I say, can you think of other ones? I think any 2 events with 2 different categories. Those are not going to be mutually exclusive. This is important because it changes how we do that or situation. So to get to there, let's talk about the multiplication rule. This is used when we have the and. So what's the probability of getting a girl and brown hair? How do we do that? Well, multiplication rule says if 2 events are independent but not mutually exclusive, then the probability that both occur together is equal to the product of their individual probabilities. So the probability of A and B occurring together is equal to the probability of A times the probability of B. Now, what does it mean by independent? It means that if we pick one item, whatever that result of that item is, doesn't affect what we're going to draw for that second item. So your daily pick 3s. 3 different machines. What happens on that first machine is completely independent of what happens on that second machine, and that's independent of that third machine, and so forth. So if they're independent, then you just multiply the probabilities to get the and portion of our probability. So here's infection of 2 parasites. We have parasite A, we have parasite B, and what I've done is said we've gone out and we've collected it, and let's say assume these are fish. Probability of having parasite A is 75%. The probability of having B is 50%. All right, so you've got a population like this that's infected with A. You've got a population that's infected with B, and then you have this slice that's infected by both A and B. All right, so if we're interested in the probability of being infected with A and B, we're looking at this slice. That probability is the probability of A, which is 0.75, times the probability of B, which is 0.5, to give us a total probability of 3/8. So 3/8 of our population is going to have both A and B. Kind of make sense? Now, these are not mutually exclusive. All right? So I ask you, what's the probability of A or B? Going out, pulling a fish, and they are infected by parasite A or parasite B. What do you think? Do we just add them?
Multiply them.
Do we just multiply them? Why can't we add them? Last time if we had the or, we added them. Okay, so they're not mutually exclusive. What would be the problem if we add them together? Well, it's over 1. Yeah, what's the problem with that? Yeah, our probabilities and our frequencies are always going to range from 0 to 1. So if we just added these up, and I chose these numbers for a reason, we exceed 1. We're at 1.25. That can't happen. So the question is why? Why did that happen? Well, it's this slice. If we added them together, we get that slice, so we've included that once, and then we add this slice together, now we've included this slice a second time, but we don't want to do that, all right? So what you do is you add them up, as you should, and then you subtract that slice. You're not subtracting the entire group, but we're subtracting it once to avoid double counting. So if our two events are independent but not mutually exclusive, we have the probability of A and B occurring together as the product of their probabilities. If we ask the probability of A or B occurring, then it's going to be equal to the sum of their probabilities minus that intersection, minus that A and B. And it's all about avoiding double counting. So here, if we did it, you'd have your 3/4 plus 1/2 minus the 3/8 gives us 7/8. So 7/8 of our population will be infected with both, or with either A or B. Some of those are just A, some of those are just B, some of those are A and B together. Not terrible, right? All right. So if we extend this to say multiple events, all right, A and B and C, the multiplication rule still applies. We just multiply the probabilities. But that assumes that order doesn't matter. So if we say the probability that we have A and B and C, it can happen in any order. And for, let's say, parasites, we don't care, what order, they were infected in. We're just looking for that. But if we're doing sequence of events, maybe order is important. Maybe we need parasite A to infect first, then parasite B to infect, and then parasite C to infect. So we'll deal with the orders. We can deal with the orders and the number of different possibilities a little bit later. So what about biology? How does this apply to biology? Well, if you didn't already pick up, genetics and our Punnett squares were basically a shorthand form for probabilities. All right? You didn't have to draw those squares. You didn't have to put your genotypes and stuff. You could have just looked at what alleles each has and then work out probability trees or probability equations to get all of those things. So what I think we're going to do is introduce the monohybrid, dihybrid crosses. Hopefully you've already seen them, as well as probability trees to help us moving forward. And we'll do that in lab. I don't see any point to kind of start that now. All right? And I'll show you those relationships. So what we'll do is we'll stop here. We'll pick up 2. And I do have probability homework. I think what I'll do is I'll probably get it posted. So if you wanted to work through it while this material is fresh, you can. You won't be able to do all of it, but at least you can get to this part, like examples like this, where I ask you, okay, you have one fish, what's the probability that it's this species or this species, and so forth. Just kind of give you practice using the addition, multiplication, and so forth. I think that's what I'll probably do. All right. See it too.